Bom, dando sequência, nós convidamos o professor Jian Ye.
So continuing, we are going to invite Professor Dian Wei that will talk about the mechanism of stabilization of greening. He's going to talk about the stabilization targeted MYC2 stabilization conferring citrus Huanglongbing resistance. He is director of the Department of Agromicrobiome and Biotechnology in the Institute of Microbiology of the Chinese Academy of Science. He's acknowledged by scientific excellence and has been awarded the award of the Excellence Young Scientist from the National Foundation of Natural Science in China. His research focused on molecular virology with innovative biotechnology, integrating artificial intelligence and biotech in the host resistance mechanisms in greening. He's published more than 70 articles as the main author, published in successful journals.
This is my great honor, a great privilege to be here, and I think that it is a very good opportunity to share our research about citrus greening in this conference, this event. Because I think that I love although it is my first time, but Brazilian people gave me a great good impressions. So I think that it will be very common to come here and have opportunity to share our progress. Okay, today my talk will be use the same topic, same title as what we published in Science cover papers. Our topic will be targeted protein stability to confer citrus greening resistance. Okay, I come from the Institute of Microbiology. Actually, so what our first idea for this, when I entered this area, when I entered the citrus greening, is culture this pathogen. Because that's the Institute of Microbiology from CAS is the biggest microbiology research institute in the world. So many people develop a new technology to culture microbials. Because that's without culturable, that it is very difficult to research. Okay, so but when I just come back from Rockefeller University and later on I'm working in Singapore for quite a long time. And when I come back to China, so my tutor, he's an academician in CAS. He suggested me to do something most important in the world in this area. So which topic? It is about the citrus greening. Why? So there's three major reasons. First is it is insect-borne disease. Insect-borne disease that totally different from what we just inoculate by ourselves, right? So similar like Francisco said, there's a whitefly transmitted virus that's very difficult to control. If without the GM technology, I think that we cannot have any solution for that. Okay, so similar like Francisco did when I do a postdoc in Singapore. So we also work with GM virus resistance. Also, this is my major target before I enter the citrus greening. Okay, so because there's no typical resistant gene so far, right? So it is very difficult to work with. And this kind of disease is not a solo disease in the world. It is so many this kind of disease exist. For example, the Liberobacter that include the CLAS. Okay, then not only the CLAS, but also you see that the spiroplasm and also the phytoplasm And also the Salmonella, a different kind of Salmonella, is very difficult to control. Those diseases, as human people, humankind, there's no way to control those diseases, especially for those trees, those woody plants that take quite a long time, and the only way for them is just to cut. Once they are identified, therefore, the 3 methods to control the citrus greening is very practical and very useful. It's similar like other diseases caused by those uncountable or difficult to culture pathogens. Okay, the citrus industry is very big, but at the first step, I didn't realize, but I think that since our paper comes out, then there will be a huge chance for us to collaborate. And many of the citrus types, comes to these slides. Therefore, it is not first reported in China. It first reported in India. Okay, so people always say that China used this as a bioweapon to invade the United States. It's not the truth, okay? So it's first occurred in India. And also the insect vector is first reported in India. And Xuefeng said that it first reported in China since 1919 in South in Guangdong province, in the coast area of, is the area? Is the area inside? Okay, so the Huanglongbing is a very big problem. I will not go detail for these slides because I think that everyone knows that it's a very, very big problem for that. But I will make you notice this tweet given by the congressman from United States, Scott Franklin. He will say that I think there's a poster in the middle of this year. He says, as chair of the House Science and Environmental Subcommittee, they are trying to exploring how AI, artificial intelligence, can generate safe and effective agrochemical innovation. Yes, exactly what we have done is just use most cutting-edge technology to help us save citrus by identifying new druggable protein or small peptide to target pathogens. Okay, comes to this slide. Let's describe why these pathogens are so different. Because, okay, as you may know, so this bacteria has no cell wall. No cell wall, that means many of antibiotics, including the ampicillin targeted the cell wall by synthesis. Therefore, ampicillin is no use for control that one. And the tetracycline can work because that it targets on the protein biosynthesis for the bacteria. And this pathogen is with very small genome, and it is living within a cell. Therefore, any drug to enter the cell is very difficult. And any peptide, if we can identify, enter the cell is very difficult. That's why this pathogen or this disease is 100 years problems for human being. That's some major problems. Okay, when I just come back to China, I'm start my own laboratory in Chinese academic sciences. I'm thinking about, because China is the original place for the citrus species. Therefore, in the early time, there's no Huanglongbing, right? It's just 200 or 300 years. So why there's no Huanglongbing in the early time? they probably have some the reasons or some resistant genes in the early time, but it disappeared in later stages. Therefore, I want to find those missing genes there. Okay, so we are trying to get some this wild citrus species, 道县野菊, yeah, this one. Okay, also this from Australia is this one. Then we do some identify not only those citrus species, but also the Lutaceous families, because the citrus belongs to this family. When you notice, I read quite a lot literature and find there's actually quite a lot of species don't have these problems. They have immune to the Huanglongbing, the citrus greening. Then it is very good starting material for us to compare the genome and compare the transcriptome and also atomic strategy to find why for example, the orange jasmine, and also a curry leaf, those plants are totally immune to the citrus greening. Okay, before I introduce our research work, I will give you a background about sensitive genes instead of resistant genes. In some cases, resistant genes and the sensitive genes are two pairs of the balance, right? The balance is the sensitive genes sometimes to negative or downregulate or fight against the resistant genes to regulate the human resistance, also human immunity, and for example, in plants, they are working in the same way. Okay, for example, here is a typical sensitive genes we call the S genes. They will be suppress the host immune system. For example, the PTI pump trigger the immunity or the salicylic acid or just amino acid. And also it will be block the entry to the host, the most topmost one. Okay, this is just to give you an idea about the sensitive genes, okay? Then the background about the protein stability is just thinking about how the host can maintain a protein level by protein degradation. Every life will have a life and a death. Every protein will also have a life, birth, and a death, okay? So when the protein finish its goal or finish his fate, it will be degraded by proteasome. That means with that, the protein level of amino acid can be reused. Once the protein finishes this enzyme, we call it the E1, E2, and E3, this relay step, and add a tag to a protein. Then once the protein add this tag with a green tag, then it can go into the proteasome. Proteasome is just like a factory to reuse the protein, okay? So that to degrade this protein as a peptide. And interestingly, when I work in Rockefeller University, my professor, Nang Hai Cha, and he is a pioneer in plant molecular biology, and he identified developed the 35S promoter. Francisco, you may know that, Professor Nang Hai Cha. And in his lab, he identifies some the PUB protein. PUB is kind of E3 ligase protein. It was always looking as sensitive genes, S gene, instead of disease-resistant gene, opposite of the disease-resistant gene, okay? Then with that, we come back to the citrus story, okay? So we do the comparative transcriptome analysis. Well, notice a very interesting trends. For example, the sweet citrus is very sensitive to the CLAS, or citrus greening, and Daoxian is kind of orange, the wild species tolerance to citrus greening. And this one, this from the Australia, this kind of immune to the citrus greening. So the resistance pattern is from the lowest sweet citrus to the highest, the Australia lemon, okay, right? This, but the gene expression of some sensitive genes, candidate, the POP21, is this pattern, okay? This is the resistance pattern, and this is the gene expression pattern. That means it is a negative trend between the resistance and the expression. Okay, so that we regard those genes as a S-gene candidate. We also tested the resistance and the gene expression in as many of the citrus genus and Lutaceous families plants. It's a very nice negative correlation between the resistance and the gene expression. That means, okay, so this one cannot work, okay? Sorry. Seems that, yeah, it's so the mouse can give a mouse. Yeah, oh, okay. It's this one is okay. You can have that. Okay, okay. I just go ahead. See that in the citrus green, in the sweet citrus, that expression level goes up more than 100. Okay, so but from the what do we see? The curry leaf, the third one from this side, is less than 5. Oh, okay. Okay, yes. Yeah, okay, this one is the most sensitive sweetest orange. These are of those 4 are totally immune to the citrus greening. So that the negative correlated, okay? This just, I want to show you that things. Okay, when we did the detailed analysis of those Lutaceous families, we identify a twin genes between the POP21 and our POP21 DN. DN means dominant negative. When we talk about the genetic items, that's what it means. This is the sensitive genes. This every citrus species contains these sensitive genes. But for these resistant genes, just only one-side mutations, okay? Yes, from this TGC change to the GCC, okay? Then the coding amino acid have a mutation. This cysteine, this adenine. This cysteine always work with the enzyme, the center of the enzymatic center. And with this mutation, then it is a natural mutation to lose the enzyme activity. Okay, with that, we are doing some enzyme activity AC. Yes, okay. So when we do this DN add-in, then the POP21's enzyme activity will be blocked. Then it actually acts as a twin genes. The sensitive genes is a brother, a big brother, And the resistant gene is the little brother, okay? Oh, okay, we take the sister also, same story. And very interesting, those 4 species contain this resistant gene, although the little brother gene, the little brother, they are all of them are totally immune or very high resistant to citrus greening. Okay, so in that case, we identify putative POP21 DN as the first resistant gene for citrus Huanglongbing. Okay, to prove that, what we need to do is, if it is sensitive genes, so when we produce a double case or double in citrus genome, then it will be more sensitive to citrus greening. If POP21 is a resistant genes, we put into the sweet orange, then it will be resistant to citrus greening, right? Okay, yeah, that is our stable transgene assay. That those is the disease-sensitive genes overexpression. That means in this plant genomes there will be 2 POP21 gene. And in those the POP21 DN overexpression lines that have a brother, big brother, and the little brother, there will be 2 genes. There's the twin genes inside in a citrus genome. Okay, here we see that if we use the salad transmission method, there will be no any citrus greening pathogen in this plant. But we use the grafting method, then we can make it happen. Yes? Okay, this method, we use the grafting method, okay? So that with that end, we can detect the pathogen, but with a very, very low level, very, very low titers. Okay, so we also have another strategy to prove that. We use the RNI. The similar strategy as our professor said is we use an RNAi and a small RNA interference to interfere this POP21 RNA at RNA level. Okay, you see that the pathogen content will go very, very low. And when we change the expression, this POP21 DN, that it is a resistant gene. So the pathogen level go down dramatically. Okay, next step, we use the genome editing strategy. You see in the sweet orange, the genome contains big brother genes, which we use the TGC and coding the cysteine. And what we hypothesis is if we can convert the sensitive genes to resistant genes, then the plant cell will more resistant to citrus greening, okay? So with that purpose, and we do the genome editing, and the efficacy is so good, so far so good. And with the genome editing, the cells, the citrus greening level go down from around 20 to go down less than 4, okay? Right, you see the further experiment to show it actually this strategy by using the genome editing to do the the resistant germplasm is possible, okay? But actually, we have generated the genome edited trees, orange trees, sweet orange trees in our lab already. Okay, so next step, we try to identify the substrate. Because it is enzyme, though we need to identify the substrate. Because it's too basic, I just go very quickly, okay? So we identify a transcription factor, which is the substrate of the POP21 E3 ligase. It is MYC2 is actually as a master regulator for host immune resistance to pathogen and also insect, for example, salad. That means it's a very good candidate. If we can increase this PAP21, the MYC2 level, then we can confer both resistance. One is against CLAS, the pathogen. Another is for the salad. In that case, we can better control the citrus screening. Yes, I will not go to the detail of those things. Another most important slide is this slide. Okay, how can we identify a peptide from our human gut? Our peptide is from our human gut, our body. Our human gut, the microbials or microbiome in our human gut, we also call it the second genome of ourselves. It is very important to keep our human healthy. Okay, so if we do the genome editing strategy, it takes, I think, in China, if we can have the products, at least 10 to 15 years. Okay, but the citrus screening is so severe right now, so we try to identify some way or some protection for citrus. Okay, so what's our thinking? This actually comes to 3 years. It's around 2022. Okay, that time, the AI, the AlphaFold 2 just come into our eyes and published in Nature. That we call the first year for the AI used for biology. And Hinton, as last year, there's Nobel Prize, both chemistry and physics, are awarded to AI strategy. Okay, so that means AI is everything. Right now, the AI integrate everything, our life, our research. We take this opportunity to use the AI to identify as much of the small peptide. Those small peptide is actually, first, they are antimicrobial peptide. Okay. Next, it is a peptide which can active MYC2-based immunity. That's two purposes. Okay. So with those aim, we develop a program. Yes, we use the anti-CLAS drug discovery progress. First, we identify as much as AMP, antimicrobial peptide. Those peptides, right now we have around 1 million from all our, in this Earth. There's any genome, any coding species, either from the microbial plants, animal, human, whatever. So we have a library, very big library, around 1 million AMP, that antimicrobial peptide. Then we have to use those protein, the second step, use the AI to find the target. Okay, so we comes to the next step, where this three. Okay, this human gut original peptide is come from the bacteria. It's a genus always step in our human gut. I would say that 100% that every people will have this species, and because it's you see that for example 50 kilogram a person, there will be contain 1.5 kilogram microbials in our gut, but almost 60 of those microbials are from bacteria, this genus. Therefore, this one we regarded as the most safety one. With that, we identify this to target the CLAS And this one is 3.3-14. The efficacy to control the pathogen is a tenfold higher, even more than tenfold than the tetracycline. That is a control, okay, the positive control. We called that apple pie, okay? Just for your easy understanding, this is called the apple pie, okay? Okay, then we learn from our gardener. He used a water bottle to drop this peptide into the vascular system. Okay, so that is very simple and a very cheaper one. We first performance 3 years ago in our greenhouse. Our greenhouse is very near to the Olympic Park in Beijing. Okay, so that after 2 months treatment, then the symptoms is reduced since 2 months, which after treatment. This picture shows that 6 months after treatment, then there is controlled plants treated with BSA, another very common proteins. Right? Then these are was small peptide treated. And look at this, the bacterial titer. Before treatment, the bacterial pathogen titer is same. Okay, so after treatment with these blue lines, you see that bacterial titer is reduced dramatically according to this one. After 5 months, it's almost very little, and to the 6 months, there's cannot detect any bacteria. Okay, actually, this data is after this 6 months treatment, we remove this treatment and keep those trees in our greenhouse. There will be 2 years after that, we also cannot detect any CLAS pathogens there. That means our peptide can really significant to inhibit the pathogen titer. Okay, not only the pathogen, but this peptide also have some inhibitory effect to the CLAD, the insect vector. Okay, so I will get this more simple. Okay, so this peptide have multiple function in not only the pathogen but also the insect vector. Yeah, similar. I will make that. Okay, we identify one of the effect that Xuefeng just introduced is the virulence factor. But this one, we identify it actually working as a lysosome inhibitor. Okay, there will be in other pathogen, human pathogen. Yes, Yersinia pestis, which called the Black Death plague. They have this virulence factor. Okay, so this is tuberculosis, TB, okay, also contain this virulence factor. But this in CLAS, this ACE5 actually has a same enzyme inhibitor. With our small peptide, we inhibit the enzyme activity, okay? So in that case, this small peptide, apple pie, can not only to reduce or inhibit the pathogen titer, but also it can disrupt insect transmissions. This work just published in Plant Communication as cover story. Okay, comes to the field test. For this apple pie, we actually did 3 years field trial, especially from the Guangxi province. This first year essay. This in the Guilin, okay. So this here is a BSA control treatment. Yeah, you can see quite a lot yellowish leaves, and this small peptide treated. It looks very nice. our teams, our students. And this also in the Nanning treatment, okay? It's the last year treatment. This control and this ampicillin treated and our small peptide treated. It's look much better than those controls. Okay, here's the details. Also, the fruit setting and the fruit colors are much better than the control. Okay, this comes to the next year, last year treated in the Chongyi is the Jiangxi province. You can see that this MYC controlled. This is our small peptide treated. Okay, so the fruit setting and the fruit color. That's also the sweet it can go up to 11 to 14. Before this MYC treated is citrus screening fruits that is only 6 to 8. This year we did another second round of experiment. This we did this control and this our small peptide treated. And this the third province testing is in the Zhejiang province, my hometown. Okay, so this MYC controlled. Okay, this APP, our small peptide apple pie treated. Okay, this Pomelo, another very big. This tree is almost more than 30 years old. Yes, it's a very old trees and very difficult to control the citrus greening. And this our this MYC controlled. This our small peptide treated. Okay, this in the Jiangxi province, another field trial. Which Giuliano and Nielsen visited in July in the greenhouse. This MYC controlled and this small peptide apple pie treated. Okay, those the fruits colors and yeah, this looks much better. Yes, but we also test. We also do the biomanufacturing, do the biotechnology improvement. You generate a small peptide from bacteria and also from the yeast. With this method, we can get a much cheaper, much easier small peptide and use for the industrialized production and reduce the cost a lot. Right now, we tested the functional of those biomanufacturing small peptide. It functions as much as good as our chemical synthesized. That means this whole industry of from the peptide identification, from the peptide biomanufacturing, and the usage of this small peptide is go very smoothly. And in that way, we can give the farmers very cheaper small peptide, okay? Yes, this is our field experiment of my PhD students and my master student. Okay, in that ways, we get a summary about our work. We first identify a gene regulatory, resistant regulatory pathways or gene road for the immune against citrus greening. And with that target, we identify, with AI assistant, the strategy, we identify a very nice, very good small peptide which can cure citrus greening pathogens. And we also test in a field trial, in I think that's a 3 years trial, and the control efficacy is higher than 82%. This year we have, and when it comes to the mechanism, this small peptide actually can have like dig a hole, just like some thrill to dig a hole in the pathogen cell, the membrane, and make the cell release their content. Okay, the pathogen will be die. Okay, you can just yeah, see this totally broken of this cell, these pathogens. Okay, this is our small peptide treated. Yeah, right now we have some patents and have been entered into Brazil. Okay, with that, will end my talk, and I will like to have questions. Okay, thank you.
Thank you so much, Professor.